Hi, so in this video I want to talk a little bit about neurons and how we came across essentially the neuron doctrine. And specifically I want to talk about how a couple of discoveries led to that. So first of all, we had advances in microscopy that allowed us to look at the brain a little better. So when you're just looking at it, a lot of times it just sort of looks, like it's not got a lot of defining features, like maybe with some other organs. The heart, you got your chambers, and it's kind of obvious what they do functionally. With the brain, it's not so obvious. So microscopy sort of helped us to see that. So with one stain called the Neisel stain, these two stains were invented early on, and one of them allowed us to see essentially the soma, or the neuron, the nerve cell in the brain, and see sort of a bunch of nuclei, and we got all these cells that were stained a nice sort of purpley pinkish color with this Neisel stain. This allowed us to see a little bit better what the brain looks like, close enough to get some detail. We also the Golgi stain was invented, and that allowed us to see like a little nerve, but also all of its dendrites, so its neurites, I guess we can call them here. Axons and dendrites are both neurites. So all these little projections that come out of it. And it didn't show as many cells in any given sample, because not all the cells ended up being stained, but it showed incredible detail of all the parts of the cells. And so this allowed people to see, well, we've got all these cells in here in the brain, and they have these really peculiar structures. And so this right here sort of enforced this idea we have of wires, nerves as wires. But this showed us how densely packed all these wires are together. So people started thinking, well, maybe there are sort of two ways we could understand this. Maybe these nerves all sort of connect together, sort of like the blood vessels in our body, and they form like a closed electrical circuit. So with electrical circuits, you have like sort of a battery right here, you got a light bulb, and then it's a closed loop, right? So I thought maybe it's a closed loop like that. Other people said, no, it's probably not a closed loop. Instead, so this is departing a little bit from the electrical model. Instead, what we've got is sort of a nerve that sends messages over here, and then when it ends sending the message, there'll be all these other nerves nearby that can pick up the message, and there's sort of a gap between them. Now, eventually we were able to tell that this is actually, with sort of advanced techniques of even better microscopy, like electron microscopy, we could tell that this is actually how things worked, that they were not closed loops, but sort of these gaps. And this gave us the neuron doctrine, and the neuron doctrine is essentially that the makeup of the brain is a bunch of individual nerve cells that meet each other and transmit messages to each other chemically, but send the messages along through the nerve cell itself electrically. So this is an electrical signal here, and this is a chemical signal. And that these nerve cells are independent. That's an important thing to remember. They're independent nerve cells. They're not all connected, sort of like tubes in our blood vessels in our body. They're independent. So the neuron doctrine is something that's going to be important as we look at sort of how the brain works and how it's structured. We look at its architecture and how it works as a tissue and an organ. And it's important because doing this right here allows the brain to do some pretty incredible things. It can have other nerves sort of like right here that might be affected in different ways, and we'll have this junction here. These chemicals can be produced and consumed in different ways that just an electrical signal wouldn't be able to. So this balance of chemical and electrical allows us to do actually some pretty incredible things with nerves and neurons.